Hey guys, this is Sarah, and welcome to Chapter 2 of the BrainFacts textbook, which we can use to study for the International Brain Bee. And I hope you guys enjoy and find this video helpful. And let's jump right in the video.
The sense organs are the brain's windows on the external world, and this is a place where there are no actual images, sounds, tastes, or smells, and instead, it's all energies and molecules. Sense organs are the key to turning these stimuli, like light waves and food molecules, into perceptions or sensations. This transformation is possible through the process of transduction, where the sense organs turn the stimuli into electrical signals, and these are then sent through a network of cells and fibers to the brain. There, the messages are processed and integrated into a seamless perception of the person's surroundings. One of the coolest senses we have is our vision. It uses a huge chunk of our brain power, around 30% of our cerebral cortex, and we've studied it a lot and know more about it than any other sense. Fruit flies and mice give us most of our knowledge of light energy conversion into electrical signals, and for higher level visual processing, we have monkeys and cats. Okay guys, so picture George Washington. Now we're going to have a zoom in into his eye. So his iris will decide how much light to let in and changes the size of the pupil after the light comes into George Washington's eyes through the cornea and the pupil. And then the lens comes in and bends the light so it will hit the retina, which is on the inside of our eye. The rigid cornea will do all the first focusing, but the lens can thicken or flatten to bring far or near objects in a better focus. So now I want you guys to imagine a mirror and George Washington is raising his left eyebrow. However, in real life, he will be raising his right eyebrow because there's a mirrored picture. And this is exactly how the retina works. What's on the right will show up on the left, and what's on the top will show up on the bottom. So it is a 2D reverse image. So imagine the retina as a sandwich. After processing by fancy specialized cells in several layers of the sandwich, the signals will go through the optic nerve to other parts of your brain for more interpretation and integration. The retina is home to 3 different types of neurons, and they all work together to send information to the brain. The photoreceptors can see light, and they are located in the outermost layer of the retina. After light enters through the cornea and lens, it goes through ganglion cells and interneurons before it reaches the photoreceptors. The ganglion cells and interneurons, they don't respond to light, but they help process and relay information from the photoreceptors to the brain, and they exit the retina together to form the optic nerve. And I forgot to mention, photoreceptors, each human eye has around 125 million of them, and these turn light into electrical signals through transduction, like we previously talked about. We have 2 types of photoreceptors, and these are rods and cones. Rods are responsible for seeing in dim light and make up 95% of all photoreceptors in humans. Cones, on the other hand, pick up fine colors and details, and I personally remember this by thinking of artists who draw cones, as they like to put fine details and colors in their artwork. Our eyes have 3 types of cones that are each sensitive to a different color range, and when they work together, we can see the whole spectrum of colors, just like a mini computer monitor, which would mix red, green, and blue to generate millions of colors. The center of our retina would have more cones, so this way our vision is clearer in the center. And there's an even smaller area called the fovea where the cones are packed really tightly together, and this is where we have the sharpest vision. The area around the fovea called the macula is critical for driving and reading, and unfortunately in many developed countries, macular degeneration, which is death or degeneration of photoreceptors in the macula, is a leading cause of blindness in people over 55. Let's talk about how these neurons receive information. In each layer of the retina, neurons receive input from many cells in the previous layer. The number of inputs varies depending on the area of the retina. In the macular region, each ganglion cell receives input from just one or a few cones, giving us sharp vision. But in the periphery, each ganglion cell receives signals from several photoreceptor cells, and this explains the less detailed and more blurrier peripheral vision that we have. If you're ever wondering how you distinguish between your coffee mug and your peanut butter jar with one glance, this is all thanks to our visual processing system. This starts with light and ends up with a clear image in our brain. When we open our eyes, light enters, bounces off objects, and lands on your retina. This is like a security guard, someone who checks the ID of each light photon before letting it in, and the ID has its own FBI agents, the ganglion cells, that tile the entire retina and provide a complete map of the crime scene. The security guard has a blind spot, the exit point of the optic nerve, but our brain has a solution. It fills up the blind spot using information from the other eye, which is having a backup. Signals travel along the optic nerve to the brain and converge at the optic chiasm, and this is like a traffic cop directing signals to the left or the right side of the brain. Finally, the information reaches the PVC in the rear of the brain. Visual information gets sent to a different part of your brain. You can think of this like different lanes on a highway, and cars will move from one lane to another, and the visual information will move from the lateral geniculate nucleus thalamus region to other parts until it finally reaches the final destination, the PVC. So basically, think of the PVC as the headquarters building. So it would have many layers and it would be densely packed. The middle layers receives messages from the thalamus, would have receptive fields similar to the retina, and preserves the retina's visual map. Cells above and below the middle layer would have more complicated receptive fields, and these can register stimuli that are shaped like bars, edges, and some higher level processing neurons. They only respond to specific objects and faces. Visual signals go into two processing streams, the dorsal stream and the ventral stream. If you see a dog running out into the middle of the street, the ventral stream would identify this dog, where the dorsal stream would make an action plan. So this is why the ventral stream is called the what stream and the dorsal stream is called the where stream. Strabismus is cross eyes, and kids with this condition have good vision in each eye, but they can't process the image from both eyes, so if they favor one eye and the other eye declines. Their vision can still be treated before the age of 8, but after that, the blindness becomes permanent. Photoreceptors that are not functioning properly can lead to a variety of blindness disorders that can be really difficult to treat, but scientists have identified the genetic defects and they can create gene therapies that recover photoreceptors. Another method would be sending electrical signals directly to the brain, which is similar to cochlear implants. Our hearing keeps us safe and it allows us to communicate with our friends and family. So whether it's our alarm clock, our music, or our guitar, there's this one common process they all share. So sound waves will reach the part of the ear that you can see, the pinna, and then they will travel down the ear canal to the eardrum. And this eardrum will vibrate. From there, the sound waves travel to 3 little bones in your middle ear, the hammer, the anvil, and the stirrup. The stirrup is like a tiny piston which pushes on an oval window, and the oval window takes the vibrations and turns them into pressure waves in the fluid of the inner ear. Finally, special cells in the inner ear, which are hair cells, turn these pressure waves into electrical signals that our brain can understand and interpret. We also have a bouncy elastic membrane like a mini trampoline inside our ear, and this is called a basilar membrane. It's a winding ramp that spirals through our cochlea. This will be tuned to different pitches along the length, and so when there's a variety of sound, so for example the speaker, the cat meowing, and then the bee buzzing, these would all be at different pitches, and so when fluid inside our cochlea starts to ripple, the bouncy ramp will start to move, and this will vibrate to higher pitched sounds near the oval window and the lower pitched sounds in the center. And we have rows of tiny hair cells, and on top of them is the tectorial membrane. And their bending action, opens up tiny channels in the stereocilia and lets ions in the surrounding fluid to rush in. The hair cells then excite the auditory nerve, and this nerve sends electrical signals to the brainstem. The brainstem then sends this to the thalamus, the relay station, and the thalamus sends this to the auditory part of the cerebral cortex. There's actually several thousand hair cells positioned along the length of the basilar membrane, and each hair cell responds strongly to a narrow range of sound frequencies depending on how far along the cochlea this location is. So basically, each nerve fiber that connects with the hair cells is tuned to a very specific frequency, and this is able to be carried over to the brain. The brainstem and the thalamus team up to figure out where the sound is coming from, the direction, and the location. The left part of your brain is responsible for speech, and if word mix area is damaged, you are not going to understand anything anyone is saying. Once hair cells die, they don't regrow, so current research is focusing on how the hair cells develop and function, and how to replace them. Our senses of taste and smell are very closely linked, and they help us navigate this world, and they are basically our perceptions of tiny molecules in the air and in our food. These can identify hazards, but they can also be exposed to our outside environment, which leaves them vulnerable for damage. Our taste receptor cells and our olfactory receptor neurons, they regularly regenerate. As we eat our food, tiny molecules are dispersed into the air, and your taste buds can judge 5 flavors: sweet, sour, salty, bitter, and umami, Japanese for savory. Tastes are detected across the tongue and not just in certain areas, and they start to fade away when you're old, but we still have 5,000 and 10,000 of these left. When taste buds are activated, they send signals through cranial nerves, and they deliver signals to the gustatory cortex and the insula in our brain. Odors float in the air, find their way into our noses on air currents, and bind to specialized olfactory cells on a small patch of mucous membrane which is high inside the nasal cavity. Axons of sensory neurons cross through tiny holes in the skull and enter the two olfactory bulbs, and there is one for each nostril. Smell is the only sensory system that sends sensory information directly to the cerebral cortex and does not pass through the thalamus. We have around 1,000 types of olfactory cells. We can identify about 20 times as many smells. This is because olfactory cells have several hair-like cilia that are subject to a number of different odor molecules, and so a specific smell will simulate a unique combination of olfactory cells, creating a distinct activity pattern that is then transmitted to the olfactory bulb and onto the primary olfactory cortex on the anterior surface of the temporal lobe. From then, olfactory information is passed to nearby brain areas where odor and taste information are mixed, which creates flavor. Research shows that people can identify odors as quickly as 110 milliseconds after their first sniff, and the size of olfactory bulbs and the way neurons are organized can change over time as well. Olfactory bulbs in these animals are capable of neurogenesis. Taste and smell are separate senses with their own receptor organs, but they are also closely related. When you're sick and your nose is stuffed, you taste things differently too. We can only taste 5 basic tastes, but smell makes it more complex. For example, sugar tastes sweeter when it's combined with the smell of strawberries, than combined with the smell of peanut butter, or no odor at all. The somatosensory system is responsible for all the sensations we feel when we come into contact with the world around us. In some hairy skin areas, some nerve endings are actually so sensitive that they respond to the slightest hair movement, and the signals from these receptors travel along sensory nerve fibers into the spinal cord, and they start their journey up to the brain. All touch information travels at the same speed, but some messages are actually transmitted quicker through myelinated nerve fibers with thick axons that are called A-beta fibers. The slower ones would be the thin, unmyelinated axons, the C fibers. Our brain has a topographical map of our entire body like a GPS system. This is located in the somatosensory cortex, and it allows us to perceive everything on the screen. Some body areas are more sensitive, such as our lips and fingertips, that are really touch sensitive, and this is because they have more touch receptors per unit area. And in contrast, the back would have fewer touch receptors and therefore would be less sensitive. Neurologists use 2-point discrimination to measure the minimum distance between 2 points on the skin that can be identified as separate stimuli. Pain is complicated because it involves both sensory and emotional components. Our sensory fibers, the nociceptors, respond only to strong or high threshold stimuli that possibly causes tissue damage. These receptors are also responsible for the burn in our mouth after eating hot sauce. During the event of tissue damage, chemicals are released which creates an inflammatory soup which amplifies the pain response. Worst case scenario would be a malfunctioning nervous system which leads to neuropathic pain, and it's like when you can't escape the pain no matter what you do. Pain signals tissue damage, and itch is caused by special receptors in your body. These are carried through small nerve fibers called A-delta and C fibers. These serve as such as like a fire alarm system. A-delta fibers responsible very fast, which is like the sharp immediate pain when you prick your finger, and C fibers are slow and steady, which is like a dull and like diffusing ache. These signals travel up the spinal cord into the thalamus, and they're sent into different parts of the cerebral cortex, the control center of the brain. Pain signals are also processed differently. When pain messages arrive in the cortex, it gets sent into the gray matter, and this is where descending pathways that can modulate pain or even send networks to induce endorphins, which is the body's natural opioids. However, the sensitivity and the efficiency of these pathways vary from person to person. Now we know that pain is actually not just one single thing, but is actually a mosaic of emotional and sensory components that influence how we perceive pain.